Today let's go over just a few very basics of vision. This will be the most basic info that you get. First of all, for the AP exam and for the Vincennes final, you will need to know the parts of the eye and kind of what they do. So just kind of a forewarning on that. Definitely pay attention to this particular illustration, and then your book goes into other stuff as well. We're going to be talking about a few things here real quick that will kind of go back to that vision or that picture. When we talk about receptors for vision, when things go in your eye, they reflect back on the retina. And the retina is made up of a ton of rods and cones. Now, the rods and the cones are the receptors for vision. Rods are basically used for nighttime vision. They do not need a lot of light to operate, but they tend to do very well at seeing shapes. But again, they don't see a lot of color. So like when you get up in the middle of the night and you're walking around, you don't turn the lights on, but yet you can still very clearly see, things laying on the floor and where the doorway is and, things of that nature, because you can see that you're using your rods. Okay? Cones, on the other hand, those are cells that are in the retina that are receptive to color. And so when you go into bright light and you see lots of vivid colors, the blues and the reds and the pinks and the oranges and yellows and so on, all of that, those are because of your cones. Cones need a lot of light to operate, and that's why in the darkness they're not very good and you don't see very well in terms of color. But cones are the color receptors. Now, you have way more rods. Let's go back to this picture here real quick. Okay, in the retina, if you take a look at the retina, it goes all the way around the back of the eye. Okay, and you have millions more rods than you do cones. You have approximately 60 million rods, whereas you only have about 3 million cones. And they're actually located in different areas. So, for example, if we look at the retina, around the edge, kind of around the peripheral area of the retina, here and then down here, you're going to have a ton of rods. Okay, not very many cones at all. Most of the cones are located right in this region, right around the fovea, which is the point of central focus. So it's kind of interesting. Sometimes we do an experiment in class that demonstrates this. An object, you have to be looking almost directly at an object to be able to correctly tell what color that object is. If things are in your peripheral vision, you can see them. A lot of times you can tell what they are, but 90% of the time you cannot tell what color they are because your cones are not picking those things up. Okay? Now, let's take a look here at the next thing, and that's the blind spot. Okay? You do have a blind spot in each eye, and I'm going to pause just for a second, and then we're going to demonstrate this. I'd like for you to just pause the playing of this video or this screencast and follow the directions on this screen to find your blind spot. Hopefully you were able to find your blind spot, and that occurs in each eye, your right and your left, and this diagram actually shows you where the blind spot's located. It's right here, okay? Basically at the point where the neurons exit the eye, there are no receptors. There's no rods, no cones in that location. So, for example, if you were looking at something close to where the word iris is here on the screen, but out here, if you were looking at something in this angle and it comes through the eye and directly goes to this particular portion of the retina, that's where the neurons exit the eye. That is the point where basically there is nothing there. Now, what is this called? This is the optic nerve. Okay, this is what sends messages back to the brain itself. Now, another thing to talk about here is with vision is color blindness. Now, the term color blindness is kind of a bad term. It's incorrect in the sense that when we think of this, we think of somebody who is not able to perceive any color whatsoever. That's typically not true. Most people can see some colors, okay? But they may have certain color deficiencies. So a better term for this would be people who are color deficient. For example, the biggest or most prevalent color deficiency is red-green color blindness. There's a ton of people who are red-green colorblind, which is somewhat humorous considering that that's two of the three colors in a stoplight. So that makes for interesting driving. But anyway, they don't see red and they don't see green as we see it. They see it as different variations of other colors or of gray or blue, things of that nature. Now, color blindness is inherited. It's one of those things that's a sex-linked characteristic. Most of the time we've already talked about this in class. If we haven't, please ask, and I'll explain how this happens. But color blindness, it's a thing that affects about 8% of men and very few women, only about 1% of females. And it is estimated that about 80% of the information you take in on a day-to-day basis comes in through the eyes. So your vision is incredibly important. Now, let's talk about one other thing, and that's called lateralization. This is kind of an interesting thing, okay? If you take a look very closely at this diagram, okay, information that's detected by the right half of each one of your eyes, the right half of each one of your eyes is transmitted to the brain's left hemisphere. So it literally crosses over. While information that's detected in the left half of each eye is transmitted to the brain's right hemisphere. And the point where the nerves from each eye meet in the brain and then cross to the opposite side of the brain, that's called the optic chiasm. The optic chiasm, it's located directly below the hypothalamus. On this diagram, you can see right here is the optic chiasm. But again, it's kind of interesting if you think about, okay, here's what you're looking at with two eyes, and the things that are to the left of your center focal point, things to the left, those go into the right side of each one of your eyes, and it gets transmitted to the right hemisphere. So again, things that are on the left of your visual field tend to go to your right hemisphere. Things that are on your right tend to go to your left hemisphere. Kind of interesting how that happens. Let's talk about a couple other things in the brain. Okay, from the optic chiasm, information is sent to the thalamus, and then it's relayed to the visual cortex. And feature detectors in the visual cortex, they're neurons that respond to different aspects of an image, such as size, shape, angles, things of that nature. Those are called feature detectors. They were found years ago by a couple theorists, or a couple researchers rather. Now, parallel processing, that refers to how that the brain processes multiple sources of information simultaneously. So when you look at this turkey, you're able to see a variety of things as well as depth and so forth. So we'll talk about depth perception in another screencast.